In this video, we will discuss about this function of this ALDH4A1. That its function is to suppress cancer cell proliferations. So this is a new paper. Here, the researchers' focus is not to find out the function of this ALDH4A1 directly, but they wanted to find out this mitochondrial pyruvate carrier, unaccounted mitochondrial pyruvate carriers, as two mitochondrial pyruvate carriers, that is MCP1 and MCP2, has been found out that can help in this TCA cycle. But there are some unaccounted mitochondrial pyruvate carriers also. So they have hypothesized that it could be this ALDH4A1 that might plays a role towards this unaccounted mitochondrial pyruvate carriers, and they also did the observations and find out that in the cancer cells they are able to see low ALDH4A1, and that could be the additional properties of this ALDH4A1, that it can not only helps in the it can not only helps in finding out this unaccounted mitochondrial pyruvate carriers, but it can also helps in the maintaining the suppression of the cancer cells proliferations. So let us now start with the results that leads to a conclusion that ALDH4A1 cause cancer cells suppressions of this cancer cell proliferation. So they use three different cancer cells, that is HEP3B, HUH7 and HEPG2. And then first what they did is they checked then knocked down this ALDH4A1, and they checked it using the Western blot. We can see that the signal goes down compared to the control, and they use different short interfering RNAs, and the results for this, the proliferations ratio to the control, is also shown that it increased in the knockdown conditions compared to the control in all the three cases. And the Western blot shows that the decrease in the panel B shows the Western blot results of the one of the cancer cell lines, which is HEP3B. It shows a decrease in this ALDH4A1 compared to the controls, and in results they have seen that there is a whenever this ALDH4A1 is suppressed, the proliferation rates are high, and whenever this in the suppressed ALDH4A1, when they transfect this wild type ALDH4A1, they see a regain of this proliferations that can be seen in the last column, compared to the case where regain of this suppression of the proliferations that can be seen compared to the case where ALDH4A1 is knocked down. At that conditions it is high, the third last conditions, and in the last conditions we can see a decrease in the proliferations when this ALDH4A1 is again supplemented. And when I did the results for this HUH7, in this case they can see a decrease in the ALDH4A1 for one of the short interfering RNAs, and for that RNAs they see a increase in the proliferations ratio compared to the control, and when the wild type is again induced, then they can see a further suppression in the proliferations. And for the HEPG2, they can see that for the both of these silencing RNAs, they see a decrease in the signals for the ALDH4A1, and whereas they see, and for this decrease in the ALDH4A1 signals, they see an increase in the proliferation ratios, and when it is supplemented with wild type ALDH4A1, they see a further decrease in the proliferations. And they did these studies in the HEPG2 cells for the P53 also. In P53 also they are able to see an increase in the proliferations with the suppression of these P53 signals, and compared to the controls, along with this P53 they also do it for this RB1, and here also they see an increase compared to the control case. In all the 4 cases, they see an increase compared to the control case. Thereafter, they did in vivo studies on the mice using the xenograft transplantations of this cells, and they can see that when the ALDH4A1 is knocked down, they see an increase in the tumor size, and similarly they see the increase in the tumor weight, and also the tumor volume increased compared to the controls. So this all indicates that this ALDH4A1 has some cancer suppression properties, particularly these cell proliferations that we have seen in the in vitro studies, and that ultimately leads to the high tumor weight and tumor size and even the tumor volume. That's all for this video. We will discuss in the subsequent videos about this ALDH4A1 role toward this mitochondrial pyruvate carriers and how it helps in these TCA cycles. So that's all for this video. If you need to understand more about this topic in detail, you can go to the reference that has been provided in the description. Thank you for your time.